Okay, now we are going to look at some of the major Calliphoridae species that you are going to find on bodies or on wounds or that are out and about. Now, I've chosen these species simply because they are the most common in North America, and they're the ones that we tend to see even in other countries on bodies first and foremost. Remember, though, that depending on where you're at, you may have to look into what species are most common. And as we start getting more and more invasive species, as we start getting people moving around and moving new Calliphoridae into new areas, these things are going to change. So really, I just did a search of the published case studies, a search of the trapping and all that sort of stuff to figure out what is there. And then I chose a couple that I know you need to know about, even if they're, say, eradicated or they're super pests and people are worried about them, just because you're going to be on the front lines and you're going to be the ones seeing when new things come in. So we're going to go through these by genera, and we're going to go by their commonality, I guess you could say. All right. So let's start with Cochliomyia. This is Cochliomyia macellaria. It is commonly called the secondary screwworm. Now, as I go through all of these, you are going to notice that I'm just saying the words. I'm saying the species. Remember what I talked about with how you say names. So these names are based on, or they're named after either major characteristics in the insect. They're named after, somebody, the entomologist who described it liked, or the person who discovered. I mean, there's a whole bunch of different ways to name these species. You will also notice that the genera tend to change. So as we get more information about the morphology, the ecology, the genetics, all of that sort of stuff, you are going to see that we get this lumping and splitting of genera. So I'm giving you what is considered the most up-to-date names in the forensic fields. You will find, as you go into the literature, as you start reading other books, that different people call things different names. And even when we have a whole group of people sitting down going, okay, you know what, we're going to lump all these together into Cochliomyia, say. There are still entomologists who are like, no, they're not. They're called this. So keep that in mind. Remember, names change. Things change around, and if you get somebody who's really just, no, it's this name, just say, oh, okay, it's that name. That's what I meant. Whatever. Now, the way to say these, when you say these words, remember what I said way back in the day, that was, you just say them like you know what you're talking about. Yeah, that's pretty much what you're going to do. You're going to sound them out and then say them like you know what you're going to talk about. And people will say things differently. So this is Cochliomyia macellaria. I say Cochliomyia, simply because I didn't want to embarrass my advisor anymore. So if you look at this word, the way that you sound it out, even there's a short O, you would sound this out as Cochliomyia, right? So Cochliomyia macellaria. And that is a perfectly fine way to say this. But this is one of the species that I spent my PhD learning about and studying and all that. And this is very, very common in this area. Most of the cases that I do from the southern region of the US has some sort of Cochliomyia macellaria presence in it. So it's very, very common. So that's why I focused on this species. Now, I was giving a presentation at, it was either an entomology or a forensics conference. I forget what many years ago. While I was still doing my PhD, my advisor was in the audience. And so I'm up there talking about Cochliomyia macellaria over and over and over again. And when you're at a presentation, there's usually a bunch of people in the room and they're listening. And then if there's questions at the end, whatever. In this particular one, there was this guy sitting in the front like to the side, and he kept making faces, sort of like annoyed faces. You know, you sort of see it out the corner of your eye when you're giving a presentation. And then he went up and talked to my advisor afterwards. Fun. And so my advisor calls me over and says, Adrian, there's a guy who wants to talk to you. And he says, you're mispronouncing the name of this species. I went, oh, really? And he goes, yeah, it's Cochliomyia. I'm like, oh, but there's a short O, and COCH is considered cochlea, right? It's phonics. And he like sort of got embarrassed, and he's like, no, it's Cochliomyia. I'm like, oh, are you upset because I'm saying cock? And then like he turned bright red, and my advisor's just like, no, Adrian. And then because I'm a jerk. I went, but it's the screwworm. What? And then he wouldn't talk to me anymore. And then I wasn't allowed to speak to anybody. My advisor was pretty pissed. So anyhow, Cochliomyia macellaria is the secondary screwworm. This is the most common Cochliomyia in North America. It is found from the southern US all the way down up to southern Canada. So it's all over the place. It is especially common in Texas, in Florida. It's a little bit into California, and we get it up north in the warmer months of the year. This, by far, is the most common species that I find on bodies, especially during spring and summer here in the South. So we see this a lot. Now, good specimens are really easy to identify, even without the use of a microscope. So if you look at this species, it is overall blue-greenish. It's a pretty big fly. But what is especially characteristic are these dark blue-green stripes on the thorax, on the notum. These are called viddi. So these viddi are present. You see these 3 stripes on its thoracic dorsal surface, and it's got these bright orange cheeks, or genna, bright orange genna. Okay, so those 2 characteristics, stripes on the thorax, bright orange cheeks, these mostly mean Cochliomyia macellaria, especially if you're in the South. Now, other more minor characteristics, the legs are kind of reddish in coloration. You've got these, remember what these are called, the callipes that are white, and they have these readily visible anterior sphericals that are usually bright white. Sometimes you see it with darker coloration. So this is pretty easy to identify in the adult. If you are out and about and you're in the South and you see a bunch of adult flies, you can just see these things landing on a body and be like, yep, Cochliomyia macellaria right there. Now, the larvae are even easy to identify. So if you look at the larvae, they're your normal smooth maggot, but they have these readily visible trachea, or these little tubes right here that are coming from their posterior sphericals. Those look like black swirly lines, which are very visible against that white maggot body. I like to call them racing stripes. They got these racing stripes down the side. So even the maggots are super easy to identify out in the field. So that makes it very easy. When you see a large maggot mass, like, oh, that's a maggot mass full of Cochliomyia macellaria. Now, these species prefer warm, humid weather, and they're most abundant during the rainy periods in the southern U.S. So we tend to see them around mid-spring, coming out as we start to get warmer temperatures. And because they like it humid, they can tolerate a lot of liquid. So we will see these in flooded areas. I've seen bodies that have been just completely saturated, so there's standing water all over them. These maggots are happily swimming through that water. They can handle a lot of water. Okay, so they really like that. Now, this species frequents carrion in both sunny and shaded locations, but we very rarely see them recovered from bodies indoors. So they're not as common indoors. They tend to be outdoors in both the sun and in the shade. Now, these are not cold tolerant. So as soon as it starts to get cold, they're going to go dormant, they're going to die. So during the warm months, they will become very abundant here in the south, and then they'll start moving up northwards. So we'll start seeing them in the northern regions, but they only last there for a short period of time because it tends to get colder really quick up there. Then they'll go dormant or they'll die off and they'll start moving south again. So they're not abundant in the northern US during the winter months. If it's an unusually warm winter, we will see them year-round here in the south. Now, this is one of the first to show up on bodies. Like I said, man, I use this more often when I'm getting bodies in the south because that's just there when they're outside. And when I see this, you can usually tell just even on crime scene photos without looking at the evidence, like, ah, yes, we got macellaria. And so that makes it pretty easy to do a time of colonization estimation, which makes it very nice. Now, very closely related to Cochliomyia macellaria is Cochliomyia hominivorax. This is commonly known as the primary screwworm, and it can be really difficult to differentiate the macellaria from omnivorax, but there is one major characteristic that makes it possible. Now, this species is infamous. This is because it causes primary myiasis. Primary myiasis is the feeding on living tissue of humans and animals. And this is a native U.S. species, but it was eradicated from the United States in 1966 because of its tendency to cause primary myiasis. When I talk primary myiasis, this is what I'm talking about. So this is a living human. The ocular orbit right here is completely infested with Cochliomyia omnivorax. So this species would show up on living people, on living animals, and just look for the tiniest openings. Either they would lay their eggs in the eyes or in little tiny wounds. So small, in fact, that we had a real problem with this species laying eggs in wounds left behind by tick feeding on cattle. So it became a real issue in the US with cattle production. As ticks would feed, they would drop off and leave. They would leave these little tiny openings, little tiny wounds, and these omnivorax would come in, exploit those wounds, and you'd get these huge maggot masses on these living cattle. It was causing massive amounts of economic problems in cattle. Humans were getting this all over their bodies. It was becoming a real public health issue. So through the 50s and 60s, scientists and entomologists worked on trying to kill off this species or eradicate this species from the US. It worked because of a sterile male release program. So this species, the female only mates once. She will mate with a male. She will keep all of that sperm in her spermatheca. And even as she does numerous oviposition events, even as she lays many clutches of eggs, she needs a protein meal. She is anontogenous. She'll go and get that protein, produce more eggs. She uses that same batch of sperm. So as she oviposites an egg, it's going to pass by the spermatheca. A single sperm will come out, fertilize that egg, and lay it in the environment. If she mates with a sterile male, that male will still produce ejaculate, but there will be no sperm, or the sperm will be sterile. So she is now trying to fertilize her eggs with sterilized sperm. So those eggs will not hatch, they remain haploid, and she doesn't mate again, and so she doesn't produce any offspring. This is how we eradicated this species. So entomologists irradiated the adults, males, and then they just released a whole bunch of males into the wild. So it became more likely for the females to mate with a sterile male than to mate with a fertile male. So these sterile males were just in such abundance that they outcompeted the fertile males. Then the females would die off without actually producing offspring, and we were able to eradicate this species. So it is currently found only in Central and South America. It is not found in Mexico. It is not found in the US on a regular basis. However, because it is native here, if it does get back into the US in some way, it can take hold very quickly. So there is an ongoing survey program to make sure it doesn't make its way back up north. So all along the Mexico and US border, there's USDA people who are surveying for this species. But the most common way that it is found is either on dead bodies or on living animals. So just a few years ago now, we actually had a reinfestation of this species in Florida. It was found by a vet. So the vet had I think it was a dog that came in with myiasis. It was a living animal, and that vet noticed that this was Cochliomyia omnivorax. So the vet called the USDA. There are procedures where you're supposed to report certain species when they come in, and then the USDA just mobilized. So there's a whole task force that deals with this particular species. They went running down to southern Florida. It had come in, I think, through somebody brought in an animal, and they hadn't gone through the proper channels. It had this species on it, and it made it through two different counties before it was fully stopped. So they were able to stop it. That was good. And now it's eradicated again. But I teach this to every group of students that I have, just so you know what you're looking at in case you see it. I want you to be able to recognize this species. Now, it looks a lot like Cochliomyia macellaria. So the overall color is usually bluish. It's got the same bright orange cheeks, but if you kind of notice, the cheeks almost look translucent when it comes to this species when compared to macellaria. Now, The primary difference between this species and Macellaria is the presence of sedal rows on the head. So they've got these dark setae in one species and not in the other. So when you get to the key, you'll be able to tell the difference. So this is Cochliomyia omnivorax. Now, one species that is also really hard to distinguish from Macellaria is Compsomyiops callipes. This is actually a species that I worked with extensively in my master's program. So it has the same dark metallic coloring, along with the same vidi, the same 3 thoracic stripes on its thorax. And actually, there aren't a lot of pictures of callipes out there, which is why I don't have any good photos for you here, because I wasn't smart enough to take them 20 years ago when I was doing my master's. Okay. And it also has these bright orange cheeks. So you can see it looks a lot like Macellaria. On the other hand, it does tend to be slightly bigger. So it is larger than Macellaria. Notice that its legs are a little more bright orange, but color is not a great way of telling species apart when you look at overall body form. The major difference is the calypters. So if you remember from the Macellaria, the calypters were fully white. In callipes, the calypters are dark with a white margin. So they got these dark setae on it with this white margin. They also have long, dark setae on the hind coccyx. Okay, so those are the major differences between Compsomyiops callipes and Macellaria. Now, callipes also tends to have a much more restricted range. They tend to be found exclusively in the southwestern United States. So we do see these a lot in rural areas in California where we do not see Macellaria. Okay, it prefers these rural areas. It also prefers very large carrion. So when I was working in California, if a body was dumped in a rural area, we tended to see callipes be the first one to show up. The problem is we don't have a lot of information about callipes because it has such a restricted range and people tend to confuse it with Macellaria quite a bit. We've done most of our research on Macellaria. I really need to get a callipes species and do some temperature data on this. All right, so that is Compsomyiops callipes. So that's the first group of species that we're going to be looking at. You let me know if you have any questions.